This video is going to help you to revise photosynthesis, specifically the light-dependent reaction and the structure of the chloroplast. So let's get started. First of all, we've got the structure of a chloroplast, which is the organelle in which photosynthesis takes place. So let's go through and have a look at the different parts. The chloroplast has this double membrane around the outside. So you can see we've got the outer membrane, the inner membrane, and the space between those two membranes is called the intermembrane space. Within the chloroplast, you've then got the stroma, which fills the chloroplast. And in the stroma, there are thylakoids, which are these little disc structures. Now, the thylakoids are surrounded by the thylakoid membranes, which are going to be really important for today's revision because that's where the first stage of photosynthesis takes place, which is the light-dependent reaction. So the thylakoid membranes form these discs. You can see that a stack of discs is called a granum, or you can say that there are many grana, which is the plural. So each stack is a granum. There are many grana in the chloroplasts. And connecting these grana are the lamellae, which are basically just folded membranes that connect those stacks of thylakoid discs. Something else that we can add onto this diagram, remember that chloroplasts have their own ribosomes. They're the smaller ribosomes, the 70S ribosomes, which are the ones you find in prokaryotic cells. They also have their own DNA. Now, together, having their own DNA and ribosomes is really useful because it means that chloroplasts can make their own proteins, such as enzymes, that they're going to need for both stages of photosynthesis. Let's just think a little bit more about the structure and how it's actually adapted for photosynthesis. We've already said they've got their own DNA, their own ribosomes, they can make their own proteins, such as enzymes. Thinking specifically about the thylakoid membranes have a large surface area, which is particularly useful for the light-dependent reaction because the light-dependent reaction actually takes place on the thylakoid membranes. Now, the bigger the surface area available for that reaction, obviously the more of the light-dependent reaction can take place. So what we can say, being a little bit more specific, the thylakoid membranes have a large surface area. On that surface, there are many electron transport chains and many ATP synthase enzymes. And both of those things are really important for the light-dependent reaction. It involves an electron transport chain. It involves the ATP synthase enzyme to manufacture ATP. So the bigger the surface area and the more of those transport chains and enzymes, the more light-dependent reaction can take place, which means ultimately the light-dependent reaction can make more ATP and more NADPH, which are the two important products of the light-dependent reaction. The other thing just to mention is that the thylakoids are surrounded by stroma. Now, the reason that's important is because the ATP and the NADPH that are made on the thylakoid membranes can really quickly get into the stroma where they're needed for the light independent or the second stage of photosynthesis. So the fact that the thylakoids are surrounded by stroma means ATP and NADPH can quickly diffuse into the stroma. There's a short diffusion distance or a short diffusion pathway, so they can quickly get into the stroma where they're needed for the light independent stage. Now, obviously, you need to be able to label the chloroplast, and you may also have to describe how it's adapted for photosynthesis. And we've focused there on how it's adapted for the light dependent stage of photosynthesis. Let's have a look at this second diagram over here. Now, this is just showing you a little bit more about how the structure of the leaf is organised for photosynthesis. So obviously we've got leaves here. Within the leaves, you have your plant cells. Now, towards the top of the leaf surface are the palisade cells. And you may remember that those palisade cells have many chloroplasts, because it's in those palisade cells that most photosynthesis takes place. They're right near the top of the leaf, so they get lots of light energy, which is required for the light dependent reaction. So those cells are packed full of chloroplasts, and those cells are also really tightly packed together, so you can get many palisade cells along the surface of the leaf cell. Inside the chloroplast, we've obviously just looked at their structure, but for today's revision, we're focusing on the thylakoids, because on the thylakoid membranes, so this image here is a real close-up of one of the thylakoid membranes. And on the thylakoid membranes, you get these photosystems, which are kind of funnel-shaped photosystems, and they contain all of the photosynthetic pigments. So they contain photosynthetic pigments, and they're the pigments that absorb the light energy needed for the light dependent stage. Now, the main photosynthetic pigment is found here at the base of the photosystem, and that region is called the primary reaction center, and it contains chlorophyll A. There are two different types of chlorophyll A, P680 and P700. They're found in the primary reaction center because that's where most of the light energy is funneled to. And chlorophyll A will absorb that light energy and use it for the light dependent reaction. Now, looking at the graph down here, you can see that chlorophyll is not the only photosynthetic pigment. You've got your chlorophyll A, of which we have the two different types, the P680 and the P700, found in the primary reaction center. You've also got another type of chlorophyll called chlorophyll B. There are other pigments which we describe as accessory pigments. Such as carotenoids, which is one example that you can see on the graph here. Now, this graph is what we call an absorption spectrum. And it basically shows us the percentage absorbance of light at different wavelengths of light. Now, you can see hopefully that these different pigments absorb different wavelengths of light. And that's a real advantage for the plants. And that's the reason they have different photosynthetic pigments, so they can absorb different wavelengths of light. So different pigments absorb different wavelengths or colours of light. You can see, for example, here, chlorophyll B has a really high absorption of light, round about a wavelength of 480. For example, that's how you read this graph. Now, looking down here at these wavelengths of light, these wavelengths represent green light, and you can see that the green light is reflected by all three of these photosynthetic pigments. And because these pigments reflect green light, this is why leaves appear green. The benefit, let's just make a note of that as well, the benefit for having a range of different pigments and absorbing different wavelengths of light is it will increase the rate of photosynthesis. Being a little bit more specific than that, it will increase the rate of the light-dependent reaction. Because don't forget, the light-dependent reaction, as the name suggests, requires light energy. As we said earlier, the main products of the light-dependent reaction, or the most important products, are ATP and NADPH. So the more light energy we have absorbed for the light-dependent reaction, the more ATP and the more NADPH can be made, which will go on to the light-independent reaction, where we actually see the formation of glucose and other organic substances. Let's get into the reaction itself. Now, if we just make sure we understand what we're looking at here, a little reminder that here's our chloroplast. We are actually on the thylakoid membrane. So we've zoomed right in on one of the thylakoid membranes, because the thylakoid membrane is the site of the light-dependent reaction. Now, just to orientate ourselves, if this is the thylakoid membrane, down here we've got the thylakoid lumen, which is just the inside of the thylakoid disc. And on the other side of the membrane is the stroma, which fills the chloroplast. And that's actually the site of the light-independent reaction, which we'll be revising in a separate video. So there are four main stages, or at least we split it into four stages to help us learn the processes of the light-dependent reaction. The first stage is the photo-ionization of chlorophyll. Now remember, chlorophyll, and chlorophyll A specifically, is in the primary reaction centre of the photosystems. Now we can see two photosystems here. We've got photosystem 2 and photosystem 1, both involved in the light-dependent reaction. We're going to be looking at non-cyclic photophosphorylation, which involves both photosystems mentioned here. So let's start with stage 1. We're going to chunk it into stages so that hopefully you can understand each one and how they all work together. So the photo-ionization of chlorophyll. The first thing to say about this stage is it involves the absorption of light. So chlorophyll absorbs light energy, which you can see here on the diagram. So you've got light, or a photon of light, striking the chlorophyll in the primary reaction centre. And to put it simply, chlorophyll absorbs light energy. Now, what's that going to do? This excites two electrons in chlorophyll. And you might think that the word excite doesn't sound particularly scientific, but that's the term that they want you to use. You're basically raising those electrons to a higher energy level. But we say that the electrons are excited. Once we've excited those two electrons using light energy, the electrons leave the chlorophyll. Now, because the chlorophyll has now lost electrons, remember, oxidation is the loss of electrons. So we could say the chlorophyll is oxidized, or because the chlorophyll has lost those negatively charged electrons, the chlorophyll is ionized. Okay, it will now have a positive charge because it's lost two negatively charged electrons. So we call that ionization. It's also an example of oxidation. Now, going back to the name of this stage, photo-ionization of chlorophyll, it should now make sense because we've used light energy to ionize the chlorophyll, hence the name photo-ionization. Okay, let's just go through that one more time. So the chlorophyll has absorbed light energy. The light energy has excited two electrons in the chlorophyll. The electrons will actually leave the chlorophyll, so the chlorophyll is now oxidized or ionized. Just to finish off this stage, where do those electrons go? The electrons are passed to an electron acceptor. Okay, and that's the end of stage one. So you can see once you chunk it like that, it makes it a lot more manageable and easier to revise and learn. Okay, now we're on to stage two, because the electrons that have been lost from the chlorophyll have now been passed to the first electron acceptor. So we're going to see now how the electron transport chain actually works. Now this stage is often called the photophosphorylation stage, because at the end of this stage, we're going to see how ADP is phosphorylated to make ATP. But obviously it required light energy at the start, so we call it the photophosphorylation of ADP. Now remember, the electrons have been passed to the first electron acceptor. And what's going to happen from that point is the electrons are going to continue to pass down the electron transport chain. And this is a series of redox reactions. Because when the electrons are passed to the first electron acceptor, that acceptor has gained electrons and so is reduced. When the electrons pass to the next electron acceptor gains electrons and is reduced, whereas the previous electron acceptor has lost electrons and so is oxidized. And that continues along the length of the electron transport chain, so we call it a series of reduction and oxidation or redox reactions. Let's have a little look at it on the diagram. Here are the electrons highlighted in yellow. They've left the chlorophyll. They're passed to the first electron acceptor. You've got another electron acceptor here, another one here. They then pass to photosystem 1. They then pass to another electron acceptor. That's the region that we are describing as the electron transport chain, because it's responsible for the transfer of electrons. Now, why is this important and why does it help? Well, as the electrons are transferred along the electron transport chain, energy is released. Okay, so at each transfer, if you like, energy is lost, which can be used in the next important stage. So as electrons are transferred, energy is released, and this energy is used to pump, which in biology always means actively transport, right? Pump protons. Remember, a proton is just a hydrogen ion into the thylakoid lumen. Let's have a look at that on the diagram. So as the electrons are being transferred along the electron transport chain, the energy is used to pump protons from the stroma into the thylakoid lumen. And you can see what that's going to do. As these protons are pumped through the carrier proteins, you can see you're creating a proton gradient where there's a high concentration of protons inside the thylakoid discs compared to a much lower concentration of protons in the stroma. So let's go back to our notes. The energy is used to pump protons into the thylakoid lumen. This creates a proton or H plus gradient. Okay, you've got this high concentration of protons inside the thylakoid lumen or inside those thylakoid discs. Because you've now got a high concentration of protons in the thylakoid lumen, the protons will diffuse back into the stroma. Okay, diffusion is the movement from a high concentration to a low concentration. So because we've got a high concentration in the lumen, they're naturally and passively going to diffuse back into the stroma. But this is the important bit. They diffuse through a channel in the ATP synthase enzyme. Okay, remember these protons have a positive charge, so they cannot just diffuse through the phospholipid bilayer of the membrane. They can only pass through the proton channels. And these proton channels, as you can see on the diagram, are in the center of the ATP synthase enzyme. Now, as they diffuse through the channel in the ATP synthase enzyme, ATP synthase can make ATP from ADP, which is our adenosine diphosphate, and an inorganic phosphate. Now that's the end of stage 2. So let's just go through it one more time. We've got the transfer of electrons down the electron transport chain. Remember, that's a series of redox reactions. But as those electrons are transferred, the energy released is used to actively transport protons into the thylakoid lumen. This creates a proton gradient whereby the protons will diffuse back into the stroma through the channel in the ATP synthase enzyme, and that allows the enzyme to make ATP from ADP and PI. Going back to the name of the stage, it was photophosphorylation of ADP. And we can see why now, because we've made ATP by phosphorylating ADP, but it required energy from light because the light energy originally excited those electrons, which were then transferred along the electron transport chain, which then allowed the proton gradient to be created, which then activated the ATP synthase enzyme. One more word for you to get your head around is chemiosmosis. This is an example of chemiosmosis, which is the same thing that you see in aerobic respiration, in oxidative phosphorylation. Chemiosmosis is basically where we are making ATP or driving the formation of ATP using a proton gradient. Okay, so it's this idea where we have created a proton gradient, and that proton gradient is driving the formation of ATP via the ATP synthase enzyme. Right, so we've now got one of the important products of the light-dependent reaction, which is ATP. We've also seen why light energy is necessary for the light-dependent reaction. But we've still got a few questions to answer, like the chlorophyll lost electrons. How do they get replaced? And what happens to the electrons when they reach the end of the electron transport chain. Now, this is where stages 3 and 4 come into play. So stage 3 is the photolysis of water. And the photolysis of water is the process that's used to actually replace the electrons that were lost by the chlorophyll right at the start of the light-dependent reactions. Now again, if you look at the name of this stage, it gives us a clue as to what it is. Photo means light and lysis means splitting. So photolysis of water is basically splitting water using light energy. Okay, so here's my water, and you can see it down here on the diagram. This is where photolysis is taking place. So light energy is going to split water. Now, if we split water using light energy, we're going to get 3 products. So the 2 hydrogens that we have are going to split into 2 protons and 2 electrons, because remember, hydrogen is just 1 proton and 1 electron, right? We've got 2 hydrogens in water. So if we split 2 hydrogens, we're going to split it into 2 protons and 2 electrons. We're also going to get oxygen. I'm going to write that as half an O2 because we always have oxygen molecules, O2, but obviously I've only got 1 of them. Another way of doing it, if you had 2 molecules of water being split, just want to make sure it's kind of balanced correctly when it gets split, you'd split that into 4 protons, 4 electrons, and an oxygen molecule, O2, okay? So that's photolysis of water, splitting water using light energy into protons, electrons, and oxygen. Oxygen is a waste product, so we don't really need to concentrate on the oxygen. We know that photosynthesis makes oxygen. We've known that since GCSE, but it isn't an important product in photosynthesis. Most of it will just diffuse out of the leaf through the stomata. Some of it may be used in respiration, but it is a waste product, so we're not going to focus on it here. The electrons are important because the electrons are used to replace the electrons lost by the chlorophyll. So if you think about that for a second, when those electrons replace the electrons that were lost by the chlorophyll will be reduced because it's gaining electrons and reduction is gain. The protons are going to help form the proton gradient that we mentioned earlier. If you have a look on the diagram, you can see the oxygen there, that's the waste product. You can see the electrons here are going to replace the electrons lost by chlorophyll to reduce the chlorophyll. The protons here are building up in the thylakoid lumen along with these other protons that have been actively transported into the lumen. So it's going to help create that high concentration of protons in the lumen, and it's going to help establish that proton gradient to drive the formation of ATP. Final stage, stage 4, is the reduction of NADP. Now, NADP is a coenzyme, okay? It's not an enzyme, it's a coenzyme, and its role is to carry hydrogen. So it transports hydrogen from the thylakoids in the light-dependent reaction into the stroma, where that hydrogen will be used in the light-independent reaction. So it's like a little carrier, it's a coenzyme. It works with the dehydrogenase enzymes and it helps to transport that hydrogen. So let's think about this. Why is this important? Well, have a look on the diagram. At the end of the electron transport chain, remember the electrons have been passed along. When they reach the end of the electron transport chain, they will be accepted by NADP. So NADP accepts electrons at the end of the electron transport chain. NADP also accepts protons or hydrogen ions, H+. So again, have a little look. The NADP, it's going to accept electrons from the electron transport chain. It's also going to accept protons that have just diffused back into the stroma through the ATP synthase enzyme. What's the result of this? Well, NADP is reduced. So NADP becomes, you can call it reduced NADP, or you can call it NADPH, because it's basically accepted hydrogen, right? It's accepted an electron and a proton, which is hydrogen. So NADP is reduced, or we've made NADPH. Now that's the end of the light-dependent reaction, because we've now seen why light energy was necessary to excite those electrons in chlorophyll. We've also seen the production of ATP using the ATP synthase enzyme and the process of chemiosmosis to drive the production of ATP. We've also seen the production of NADPH, or reduced NADP. Those are the two important products because they will go into the stroma, or they'll diffuse, to use a more scientific term. They'll diffuse into the stroma because they are needed for the next stage of photosynthesis, which is the light-independent reaction. Remember, we have also made oxygen in the photolysis of water, but oxygen, as we said before, is a waste product. It's not used in the second stage. These are the two important products. Now, the light-independent reaction, as the name suggests, doesn't directly need light energy, but I'm sure you can appreciate that without light, the light-dependent stage would slow down and stop, which would mean there'd be no production of ATP or NADPH, which ultimately means the light-independent reaction would slow down and stop because it wouldn't have the ATP and the NADPH that it needed to continue. So even though it's called light-independent, it does rely on the products of the light-dependent reaction, and it would stop in the dark. I'm just going to go back to the previous slide so you can see the titles of the 4 stages. So learn them in order and learn them in detail. We have the photo-ionization of chlorophyll, which is basically the absorption of light. We have the photophosphorylation of ADP, which is where we're making ATP. Again, it used light energy and it required the electron transport chain. We then have the photolysis of water, splitting of water using light energy. And finally, the reduction of NADP to produce NADPH. Make sure you know what the 2 important products are, and also make sure you understand where exactly it takes place. So this has all been on those thylakoid membranes, which have a large surface area for more of this reaction to occur. More electron transport chains, more ATP synthase enzymes means more ATP and more NADPH. The next video that we're going to be doing is on the second stage of photosynthesis, where we'll look at the light-independent reaction, also known as the Calvin cycle or the dark reactions.